welcome to this in our series of Ecological Continuity Trust webinars. My name's Keith Golding. I'm the chair for this session, and we're delighted to welcome Richard Bardgett to speak to us today, and all of you, of course, who are joining in. Just some housekeeping. If you could keep your videos and sound off, please. If we do want you to actively join and ask a question, we'll ask you at the end, but please use the chat facility to set your questions. Our Executive Director, Ben Sykes, will keep an eye on the chat facility, and he will compile the questions at the end and put them to Richard. But welcome everybody, and again, we're delighted to welcome Professor Richard Bardgett. This is a summary of his CV, which is very long and distinguished. So Richard is Professor of Ecology at Manchester University, where he's been since 2013. He graduated from the University of Newcastle upon Tyne in 1987 with a degree in soil and land resource science, and then moved to Lancaster University, where he gained his PhD in 1991. He's published over 350 papers and several books, including the most recent, which was Earth Matters. He's a highly cited researcher in ecology and environmental sciences, and he was elected a Fellow of the Royal Society of Biology in 2011. He's a founder member of the Global Soil Biodiversity Initiative, and he contributed to the United Nations Intergovernmental Technical Panel on Soils report, the status of the world's soil resources, in 2015, the UK Government Soil Health Inquiry in 2016, and the Climate Change Risk Assessment in 2017. He was also a coordinating lead author of the UK's National Ecosystem Assessment in 2011, and he served as president of the British Ecological Society from 2017 to 2019. On a personal note, I thank Richard for his work as a member of Rothamsted's Board of Directors for 10 years. We were very pleased with all that Richard contributed. His research interests are summarised as plant traits and ecosystem processes, soil biodiversity and ecosystem function, plant-soil interactions and climate change, and herbivore impacts on terrestrial ecosystems. Today, he's going to talk to us about the research at Colt Park Meadows, which is overseen by the staff of the Soil and Ecosystem Ecology Lab at Manchester. So Richard, over to you. Just again to ask all participants, please, to turn off your videos and turn off your sound. And if we do want you to join us actively, we'll ask you to do that later. But Richard, over to you. We're looking forward to hear what you have to say. Thank you.
Thanks very much. I'll just bring up my presentation. Is that all clear?
Yeah,
slide?
that's perfect. Well done. Yeah.
Thank you very much. Well, thank you very much for that introduction. What I'm going to talk about today, as Keith said, is basically about Colt Park Meadows. And Colt Park is an experiment, a long-term experiment. It was first set up in 1989. Personally, I got involved in the experiment about 1993, and I'll say more about that later. And basically, it's an upland mesotrophic grassland. And the main goal of the experiment, when it was first set up, was to restore botanical diversity within what was an agricultural improved meadow. Just in terms of where it is, I guess many of you in the audience might know about the three peaks in Yorkshire. So there's Penigent, Whernside, and Inglebrough. And Colt Park, if you've been up any of those mountains, or you've been close to Ribblehead Viaduct, which is in the middle figure here, or the middle image here, you will probably have got very close to Colt Park Meadows. And it's right in between all those three hills, Whernside, Penigent, and Inglebrough. And it's very close, probably about a mile from the Ribblehead Viaduct. So many people might have been close to the experiment, but not know that there was this particular long-term experiment at the site. As I said, the experiment was actually set up in 1989. And the person who actually set it up, I don't know if you can see my cursor, but there's this gentleman with a cap on in the background. I did try and find a picture of Roger, but this is Roger Smith. And Roger set the experiment up in 1989. And actually, Roger was a lecturer of mine at Newcastle University. Keith mentioned I did a degree at Newcastle. Well, Roger was actually one of my lecturers while I was at university, which is how I actually got to be involved in the experiment in the first place. There's quite a few people here. I'll just mention a couple of others who've been involved from the onset. This is Peter Corkill, and Peter was at the time the warden for the Colt Park Meadows site, which is part of Inglebrough National Nature Reserve. And one of the things I would emphasise throughout the talk is that none of this would have been possible without the support of Natural England. And particularly Peter, when he started the experiment with Roger, was a great help in ensuring the establishment, but also the maintenance of the experiment. Just a couple of other people just to highlight very briefly. There's Val Brown, who's on the right-hand side, and Val has also been a big supporter of the work that was done in these meadows. And Val at the time was an advisor for DEFRA in their Species Rich Meadows program, which was one of the main funders of this particular experiment. And just the one other person I'll mention is at the front here. This is Juliet Frankland. And just to show how sort of close all this experimental work was, Juliet was actually my PhD supervisor, was again someone who got me involved in these kind of experiments. So that's something about the actual people. And I think one of the sort of fascinating things about the Colt Park experiment is the people who've been involved, which have changed over time, but also the focus of the research has very much shifted over time, mainly driven by scientific, but also policy relevance. As I said, it started in 1989. And at that time, the primary focus of Colt Park was on restoring botanical diversity within agriculturally improved grassland. That was the main focus. And this experiment, as I'll say later, was supported by DEFRA with that primary goal. But then it started to shift. So at the turn of the millennium, around 2000, we were funded again by DEFRA in a large project called Dig4. And the focus there was on the role of plant-soil interactions in facilitating botanical diversity restoration. And then gradually, this focus of the experiment shifted. So around 2005, we started to work on soil carbon sequestration. Around 2010, we started to work on ecosystem service multifunctionality, again funded by DEFRA. And at present, and I'll say more about this later, we're working more on resilience and how restoration management can impact or potentially benefit the resilience of ecosystem services and functions to environmental change. So what I want to do in this talk today, it's a relatively short talk, so I can only really touch on some of the work that's gone on. But what I'd like to do is really give a sort of historical perspective of the work that's been done at Colt Park right from its onset to present. And I'm really going to focus on three particular themes. I'm going to start by talking about the initial work on restoring botanical diversity and how I actually got involved in the experiment. I'm then going to focus on soil carbon sequestration, which was a major focus, as I said, from about 2005 to 2010. And then I'm going to talk about ecosystem multifunctionality and also resilience, which is really the sort of main focus of the research that we're doing at present. I should also say that this is a picture of the experiment here. This is actually quite a rare picture in that it's beautiful blue skies, which in that part of the world is actually quite rare. I was just up there earlier this week, and I can tell you that it was very different in terms of the weather conditions. It's about 1,000 feet, and the weather in that area can be quite extreme at times. So to start with, to say a few words about botanical diversity. Well, when the experiment started, the design was actually quite complicated in that there were 4 different treatments involved. So this is the experiment that was set up by Roger. I wasn't involved at this particular time, but it had 4 different treatments. And as I said before, the main goal was to enhance botanical diversity of what was an agricultural improved upland meadow. So prior to the experiment, it had relatively high grazing pressures, it would be fertilized, dressings of lime, for example. And it was a classic lowland, relatively species poor grassland, what would be classified as an MG6. And the experiment, as I said, was actually quite large. There were 3 levels of grazing, So there was autumn grazing with cattle and sheep, spring grazing, but also both. There were 2 fertilizer levels, actually quite low levels, 25 kilograms per nitrogen, so very low levels generally for nitrogen addition to grasslands, but there were 2 levels, none and also 25 kilos. 3 different cutting dates, early, mid and late, so 14th of June, 21st of July and September, and 2 seed addition treatments, about 19 different species, sourced from commercial suppliers, but also locally sourced species. So it was quite a complicated experiment, and I guess the goal at the time was to identify the combinations of treatments that would be most successful in terms of restoring botanical diversity. Now, one of the first papers that actually emerged from the study on botanical diversity wasn't a study that I was involved in. This was one that Roger Smith published in 2000. And I think that reflects the fact that actually it took an awful lot of time to actually detect changes in plant diversity. These data were 8 years after the establishment of the experiment, and it took 8 years to see any significant changes in plant diversity, and they were nowhere near at the time the actual target community. As it says there, it actually took 14 years, so beyond this actual publication, to get anywhere similar to the target community, which was a traditionally managed hay meadow, an MG3 community, an anthox, anthem, geranium type meadow, which was quite prevalent in that area. And the data here really sort of show the treatments and the combinations of those treatments which were most successful. And I guess the sort of headline message that came out of this particular study was that the largest increases in plant species richness came with actually a combination of treatments. So autumn and spring grazing, a mid cutting date, so relatively late cutting date, 21st of July, and also seed addition and fertilizer. So really, the most successful treatments relied on all 4 of those interventions in order to boost plant species diversity within those systems. And as I said before, they still weren't close to the target MG3B community. And that took about 14 years, at least in terms of composition. Another thing just to emphasize, and I'll touch on this later, is that the lowest yields were associated with autumn and spring grazing, an early cutting date, and no fertilizers. So some of the treatments that were best for diversity had a trade-off in terms of yield, in that the lowest yields were associated with autumn and spring grazing and also no fertilizer addition. Now, I actually got involved in the experiment in 1993. And at the time, I was relatively young, and I was working on hay meadows in the particular area. This is actually one of Juliet Franklin's meadows in Ravensdale, where I was doing some work on microbial communities in differently managed grasslands and how you can relate that to the changes in diversity that you see above ground. So the particular focus of my research at that time, I was only just out of my PhD and then through my first sort of research positions, was looking at how plants within grassland communities can modify the soils and their microbial communities, and what the feedback consequences were for the growth of plants. I didn't have any funds at the time, so that's why that's actually my daughter, my eldest daughter. She's actually 29 years now, and I took her soil sampling at the time. And it's interesting that she actually didn't go down the route of science. She went down to art school rather than pursuing the science of soil. But anyway, the general model that I was working on at the time was the idea that management intensification in sites imposes large changes in the microbial communities, and that those shifts have consequences for nutrient turnover, but also plant microbial associations that could benefit the diversity of plants. And I did sampling in lots of different locations, including meadows, and the general model that we were testing was the idea that more intensive systems have these bacterial-based food webs, which are related to very rapid cycling of nutrients, high rates of nutrient loss, low mycorrhizal infection. So mycorrhizal fungi associate with roots. So basically, there's very low potential for plant microbial associations that can promote the diversity of plants. So what we proposed on the basis of this, there was that traditionally managed systems, the hay meadows that you find within Yorkshire Dales and other areas, they actually had these more fungal-based food webs. And they were associated with slower rates of nutrient turnover, high rates of mycorrhizal infection, which are known to promote plant diversity, and also high potential to promote plant diversity through a range of different mechanisms. So the general idea I was working on at the time, independent of Colt Park, was this idea that this could impose a constraint on biodiversity restoration. So any intervention that could increase the abundance of fungi relative to bacteria could potentially accelerate or facilitate the process of biodiversity restoration. So I got in touch with Roger at the time. This was around 1993, 1994. And I went to Colt Park and took a whole load of samples over time. And these are some data that came from actually 2000. So we sampled the sites early on, I think about 1994. We did them in 1996. We found absolutely no treatment effects whatsoever on the microbial communities. But then by 2000, we started to see responses. So the measure we have here is what's called the fungal to bacterial PLFA ratio. So that's really just a crude assessment of the relative abundance of fungi and bacteria. So as I said before, an increase in the relative abundance of fungi is more associated with these extensive species-rich systems, whereas if you increase the relative abundance of bacteria to fungi, that's more akin to an intensive system with less opportunity to actually promote plant diversity through plant microbial interactions. Now, what this slide shows here on the left-hand side is the number of plant species, and around this time, particularly around sort of 1998, 2000, there started to be clear signs of certain treatments having strong effects on plant diversity. And in particular, the absence of fertilizers and seed addition had a strong positive effect in comparison to the addition of fertilizers with no seeds. And when we looked at the microbial communities, we saw a similar kind of pattern. Fertilizer addition had very low or relatively low ratios of fungi to bacteria. The removal of fertilizers, particularly with the addition of seeds, so these are the 19 seed species that were added to those communities, which were mainly slow-growing plants, actually boosted significantly the ratio of fungi to bacteria. Now, there are many reasons why that could occur, and what we were interested in at the time was whether that was driven by individual plants introduced into the community, or whether it could be other factors, for example, changes in organic matter quality or soil abiotic conditions. But we got a reasonably good amount of evidence to point to the fact that the introduction of particular plants into the community, which had sown, were responsible for, in part at least, shifting those changes in the microbial communities. And there are just two pieces of evidence here. This is a PCA graph. Roger was very fond of these principal components graphs. And I guess what this one shows here is that fungal PLFA, or fungal biomass, the ratio of fungi to bacteria here, were associated with seed addition treatment, in particular with legumes, but also with stress tolerator species. So these are things like Plantago lanceolata, Ranunculus ancris, Prunella vulgaris, Leontodon hispus, etc. So at least in terms of correlations or associations across all the treatments, we started to see these relationships occurring. I had a PhD student, Louise Innes, at the time. She also did some experiments at Colt Park in individual plots where she sowed in monocultures. And again, we saw that there were certain species, particularly these legumes, so this is Trifolium potens, Plantago lanceolata, Achillea millifolia, for example, here, they had a strong positive effect on the ratio of fungi to bacteria in the soil. So really these kind of findings got us thinking about whether the changes in the soil as a result of the introduction of plants could have beneficial effects on plant diversity through a range of feedback mechanisms. Now, we did a range of experiments on that, and I haven't got time to go into too much detail, but I just want to identify one experiment that we did relatively recently, which was only published about 3 years ago. And this, I think, provides clear evidence that changes in the fungal communities in particular, as a result of changes in the plant community in the presence of individual plants, has feedback consequences that could potentially result in changes in plant diversity, positive effects on plant diversity. So what we did here was basically grow a whole range of different plant species present at Colt Park within individual soils, over 3 growing seasons, and then we looked at the bacterial and fungal communities. The data here are really just the fungal communities, which are separated into pathogens, mycorrhizal fungi, root-associated fungi, which are known to benefit plant growth, and also saprotrophs, the decomposers. The majority are actually saprotrophs, but what we found was across all these species, there were 14 species we grew in this particular experiment, there was strong divergence in the relative abundance of beneficial organisms, mycorrhizae, but also those which could be detrimental, i.e. pathogen abundance. So we could see that individual plants do select or have quite strong impacts on the abundances of these beneficial, but also these detrimental organisms. What about the feedback consequences for plant growth? Well, we did a range of feedback experiments. These are experiments where we grow plants in their own soil, the soil they've actually cultured over 3 years, compared to their growth in the soil which has been cultured by another species. So generally, a negative feedback would suggest that they perform worse in their own soil, and that response is due to the effect they've had on the organisms that live in the soil. And what we actually found was across all those species which had a positive effect on the diversity of mycorrhizal fungi in the soil actually had a positive feedback. So the greater their impact, the greater their positive feedback on their growth. So if they change the conditions in the soil in terms of benefiting mycorrhizal fungi, the general idea there is that those species will benefit them in future generations. Whereas those species which encouraged pathogens, perhaps unsurprisingly, had a strong negative effect on their growth in subsequent generations. We also found that when we looked at what type of plants have these positive or beneficial effects or negative effects, and we found that basically fast-growing plants with high nitrogen, fine roots, things like Lolium perenniae, for example, generally accumulate fewer mycorrhizal fungi, but they do accumulate these more diverse pathogen communities, resulting in net negative interactions. So these evidence here, they're not directly related to the changes that occurred at Colt Park, but what they do suggest is that changes in the soil communities resulting directly from the plants that are present in those systems could induce feedbacks that could facilitate vegetation change and potentially increases in plant diversity in these restoration experiments. Now, as I said, the focus of the experiments has shifted over time, and around 2000 was when we were starting to see these quite dramatic effects on plant communities, but also these significant effects on the organisms and processes that occur within the soil. And the actual design of the experiment also changed. What we did then was actually simplify the design. So we removed the grazing treatments. If you remember, before, autumn and spring grazing was considered to be the optimum in terms of plant diversity along with the other treatments. So we removed that, and we also removed the cutting date treatments. So we only retained the plots that were, or the blocks that were present within the sites which have been most beneficial in terms of grazing, but also cutting dent. So that left us with 2 treatments, NPK addition and mixed seed addition, but also we introduced 2 other treatments, farmyard manure, so the addition of manure, about 12 tons per hectare each spring, and also we introduced a clover addition experiment. And that was really off the back of several lines of evidence suggesting that Trifolium pratense, or red clover in particular, seemed to have a strong effect on the properties of the soil. So we wanted to study this in more detail. And the experiment was changing not just in the science, it was also changing in the people who were involved in the experiment. And just to note, we got a funding from BBSRC at this time to look particularly at how the treatments impact on carbon sequestration and the mechanisms involved. I also teamed up with a colleague of mine, Nick Hossel from Lancaster University, who was a specialist in measuring greenhouse gases. And you can see in this figure here, these are the mobile laboratory facilities that we used at the site to measure greenhouse gas emissions from the different treatment plots. So the general idea that we were testing here is sort of presented within this graph here, which is a sort of general framework that we were working towards in this particular project. And it was the idea that the actual traits of the plants within a community have impacts on the microbial communities or the associations with the microbial communities, for example, the mycorrhizae, the symbiotic organisms, but also the bacteria and fungi I mentioned previously. And generally, it was the idea that plant communities dominated by more slow-growing species that produce recalcitrant litter would lead to shifts in the community more towards these fungal-dominated systems with a greater abundance of symbionts. There'd also be a greater investment in roots compared to these faster growing species that promote the bacterial communities. So it's very similar to the model I presented earlier, but it was more refined in terms of our increasing understanding really of how plants and microorganisms associate with each other and how they impact on soil processes. So the general idea here was there's a shift in this direction towards these fungal dominated systems with symbionts. The conditions would be more conducive to carbon sequestration. So we went out and tested this in the Colt Park experiment. And I've just got a few slides here just to briefly show you what we found. We actually found was the legume treatment in combination with those treatments that were most effective increasing plant diversity, i.e. the removal of fertilizer and the introduction of seed. It was those treatments when Trifolium pratense was added that had a strong, significant effect on the sequestration of carbon, but also of nitrogen within the soil. And the rates of sequestration were actually quite high, around 300 grams of carbon per meter squared, which is actually an extremely high rate of sequestration. It did stabilize after that, but there was an initial increase, which we think was predominantly down to the roots and how the roots impact on the soil system. We did look in some detail at the actual mechanisms involved. And what we found was that in the presence of Trifolium pratense, there was an increase in the amount of carbon within the fine fraction or a mineral associated fraction within the soil. We also did some CT scanning. Ian Young, who was at Dundee at the time, was involved, and we took cores and did CT scanning of the soils. And what we found was the presence of Trifolium within the plots at Colt Park, where it had been introduced, also increased aggregation in the soils, which could in turn lead to physical protection of carbon within the fine fractions. We also find suppression of enzyme activities involved in decomposition of organic matter. So all these things together, we think, contributed to this buildup of carbon within the soil. The final line of evidence, I guess, that made us more convinced in terms of what was actually happening, because it's only just one location and one experimental system, but we also looked at greenhouse gas emissions across the treatments over a year, from January right through to December. And what we found, just looking here at gross carbon or respiration from the soil, so this is root and also soil respiration, in the presence of Trifolium pratense, rates of soil respiration were reasonably consistently declined, particularly during the spring and early summer months. So we think in these experiments that the introduction of a single plant species at relatively low amounts of cover, in this case Trifolium pratense, can have disproportionate effects on soil processes, in this case leading to an increase in the sequestration of carbon contained in the soil. So the last area that I'd like to talk about is more the kind of work that we're doing at present. And I think this kind of work really reflects a growing awareness that grasslands and other ecosystems actually provide a range of ecosystem services. So not just carbon sequestration or yield, for example, they can present a whole range of different benefits for society. Water retention, climate relegation, soil fertility maintenance, pollination, the list goes on and on. It's also, I think, there's a growing recognition that grasslands actually do play a crucial role in supporting biodiversity, ecosystem services and human well-being across the globe, but that globally grasslands are under threat as a result of grassland degradation. So there's sort of growing awareness, societal, but also policy relevance of the importance of grasslands and the need to manage grasslands in a more effective way towards the production of multiple ecosystem services. So not just focused on yield or not just focused on carbon sequestration, focused on delivering a broad range of ecosystem services. And really this became the focus of the Colt Park experiment. During the sort of early 2000s, we got more support from DEFRA for a 5-year program, which was looking at multifunctionality with a focus on carbon sequestration, but also the trade-offs or benefits or associated synergies with other ecosystem services. The people involved also changed. So we had Sue Ward was the person who was employed on the project. Andy Wilby from Lancaster was involved, who was working on pollinators. The new warden at the site, Colin Newlands, was also actively involved in this particular experiment. So what we did here was not just measure carbon storage and uptake, as I showed you previously. We also measured a whole broad range of other different properties using data that we collected over time. So we had data on nutrient retention, the amount of nutrients contained in soil or the leaching of nitrogen out of the soil, and also greenhouse gas emissions. We also looked at pollination services. So we measured flower availability and visitation across the plots. And we also measured production indices, hay yield and floral diversity. And then we used all these data to measure multifunctionality as an index measure. So multifunctionality is just really a measure of the simultaneous performance of a broad range of different functions. So in this case, we had production related, diversity, pollinator related, carbon, etc., etc. And then we could measure the response in terms of the effect, standardized effect of the different individual treatments on the individual services, but also on multifunctionality. And what we found was, perhaps as you'd expect, the individual interventions had strong trade-offs in terms of the different ecosystem services. The most obvious here is nitrogen addition, increased yield, reduced plant diversity, reduced plant pollinators, and also increased the amount of nutrients lost from the system. Unsurprising results in terms of knowledge about how high use, well, fertilizer use would impact on these processes. Farmyard manure addition also had similar kinds of trade-offs, but perhaps less strong in terms of nutrient leaching and effects on soil properties. We also looked at the other treatments. These are more ecological interventions, so mixed seed and red clover addition. They didn't have massive effects on these different services or multifunctionality, but what they tended to do was neutralize those effects. So they had positive effects on diversity and pollinators, as you might expect, but generally neutral effects on other services like nutrient retention and carbon storage. So they had relatively small effects, but they did tend to minimize the effects on multifunctionality or the trade-offs associated with the different types of measures. Now, we've recently explored these data in more detail. It was actually Shang-Chi Liu, who's a postdoc who's working with me at present. And what he's done is take all the data from that particular piece of work and additional data and tested whether the number of management interventions can actually minimize those trade-offs and increase ecosystem service multifunctionality. Now, these are unpublished data. They're a very recent analysis, but what they show is if you look at the multifunctionality index, so these are the control plots, these are the single interventions, these are with combined 2 interventions, 3 interventions, and 4. And you can generally see that there's an increase in multifunctionality with a greater number of interventions. And this really sort of mirrors what we found in the initial experiments in terms of plant diversity, at least, that to enhance diversity, you need a combination of treatments, no fertilizer, seed addition, and changes in grazing regime. And it seems to be the same case here in terms of multifunctionality, all those range of different properties, that the more interventions are combined, the greater the positive effect on multifunctionality. Now, I've just got a couple of slides to finish off on, because I realize I probably talked longer than I should, but I just want to say something about our current focus, which really sort of builds on that particular results, but also different aspects of it. Because one of the things that we're particularly interested in now at Colt Park is resistance and resilience, or what we might call stability. In other words, the capacity of the system, the different management regimes, to resist and recover from perturbations, in particular things like extreme climatic events. And these are just some data. This is from Adam Bilton, who's actually a PhD student working at Colt Park now. And he gave me these data yesterday. So they really are hot off the press. And essentially what he did was measure the stability of respiration and net ecosystem exchange, which are key measures in terms of carbon cycling, among other measures. These are just a snapshot of the data. And he measured them over 5 months at 2 weekly intervals. So you can measure the temporal variation in these measures as an index of the stability of the system. So the less variation, the higher the stability. But he also introduced a drought during the summer months, a 6-week drought in terms of simulated drought using rainfall shelters. And you can see in these experiments here, firstly in stability, the higher the value, the more stable. These are the most successful treatments in terms of diversity. And you can see that actually they're very stable in face of drought. They retain the same stability in terms of respiration and also net ecosystem exchange as does the other treatment in the control. But in the treatments that were least successful in terms of plant diversity restoration, so fertilizer with no seed, these are the controls effectively, you can see that under drought, they were less stable than were the actual controls. So this suggests that these treatments that have been more effective in restoring botanical diversity and ecosystem multifunctionality and other properties have also increased the stability of these two measures, at least in terms of their capacity to withstand a simulated climate extreme. And we're now looking at this in more detail. This is Tian Yang, who's also doing his PhD at the site now. We're using various pulse labeling approaches to look at the transfer of carbon through the system to try and get an underlying understanding of the mechanisms involved in terms of the stability in terms of carbon flows through the particular system. So I'm aware of the time, so just to wrap up in terms of key messages, I think that the first thing I would say is that, you know, having been involved in this experiment now for nearly 30 years, which I find quite frightening myself, actually, in terms of thinking about it, it's been really quite a journey in terms of the science, but also in terms of the people involved. And it's been really quite an evolution, really, in terms of the people, but also the actual scientific questions that we've actually addressed. I mean, in terms of the scientific methods, I think one key thing that we have discovered here is that restoration takes a lot of time. As I said, it took 14 years to get anywhere near the actual target traditionally managed meadow community. But I think what we've also gained is mechanistic insight into some of the mechanisms involved. We've also demonstrated some additional benefits of grassland diversity for ecosystem multifunctionality and individual functions and minimizing these trade-offs. And I think the other thing, as we're working on now, is we're starting to get some indications, and I stress just some indications because we've got a lot to do, in terms of those treatments which seem to be most successful for biodiversity restoration and also for multifunctionality, also seem to confer stability in terms of their resistance to things like climate extremes. So just to finish, in terms of acknowledgements, I think I have to obviously acknowledge Roger, because without Roger, we wouldn't have the experiment, and I certainly wouldn't be involved. Andrew Hind, he's the current in charge of the actual site, Colin Newlands and Peter from Natural England. Again, without their help, this experiment wouldn't have been possible, and they still provide an excellent service for us in terms of maintaining the experiment. The other thing, Richard Brandhardy, Steve Peel and Val Brown, who've all been amazing supporters, I would say, of this particular experiment. And again, those early experiments that we did wouldn't have been possible without their support. And a whole list of different people. I've named a few other people, but there have been many other people who have actually been involved. So on that note, I'll finish because I'm aware that we're running out of time. So thank you very much.